Welcome to Module 2, where you'll be learning about heredity, prenatal development, and birth. As you learn about heredity, you'll be learning about genes and genetic influences on development. As you learn about prenatal development, you'll learn about the stages and milestones of prenatal development. And then you'll be learning about the birth process, and specifically, you'll be looking at the main events and assessment of the newborn. By the end of our first section on heredity, you should be able to define what genes are. You should also be able to distinguish between mitosis and meiosis, genotype and phenotype, homozygous and heterozygous, and dominant and recessive. You should also be able to describe some genetic disorders due to a gene defect or chromosomal disorders. You'll also be able to define polygenic and incomplete dominance and describe the function of genetic counseling and why some people may seek genetic counseling. You should also be able to define behavioral genetics and describe genotype environment correlations and genotype environmental interactions, and then define what epigenetics is. Genes are recipes for making proteins, and proteins affect the structure and function of cells. Humans have about 20,500 genes, and these were identified in a project called the Human Genome Project. Genes, as most of you know, are located on chromosomes. Let's take a look at more of the basics of genetics. As many of you know, humans have 46 chromosomes, and you can see an image of this on the screen below. We have 23 pairs, and the first 22 pairs are called autosomes, and the 23rd pair would be our sex chromosomes. And one member of each of these pairs is inherited from each parent. After conception, most cells are created by mitosis, and you can see mitosis at the top of the image that's on the screen. The cell's nucleus makes an exact copy of all of the chromosomes, and then it splits into two new cells. Gametes, which are the reproductive cells, the sperm and the ova, they're formed by meiosis. A gamete's chromosomes duplicate, and then they divide twice. This then results in 4 cells containing only half of the genetic material of the original gamete, as you can see in the image on your screen. So each sperm and egg possesses only 23 chromosomes, and then they combine. As you know from your reading, we all have what is called a genotype, and these would be all of the genes that we inherit. We don't know about all of the genes that we inherit because not all of them show up physically or behaviorally. Only some of them do. And so we have what's called a phenotype, and these are the features that are actually expressed. So, for example, this could include our physical features like our eye color or the shape of our earlobes, or our internal traits like our blood type or diseases. And many of these are what we would call polygenic. They're influenced by several genes. I remember many years ago when I was in biology in high school and even in college, I remember learning about how genes were passed on from parents to their children. And I remember learning about something called the Punnett Square. That might be something that some of you are familiar with, where you could estimate the probability that, let's say, for example, 2 parents who had a child would have a child who had blue eyes or brown eyes or green eyes. And so it seemed so simple when I was learning about this, but actually it turns out that eye color is not that simple. Eye color is an example of polygenic inheritance. We don't just have one gene for eye color. It actually is thought to be influenced by up to 16 different genes. And so we would say that this is a polygenic inheritance that's taking place. Many genes are influencing this trait, not just one gene. Additional examples of polygenic inheritance include skin and hair color, height, the risk for certain diseases. So some diseases are polygenic, the resistance that we have for some diseases, also intelligence, even blood pressure, bipolar disorder, autism, longevity. So lots of traits are polygenic. Alleles are copies of genes that influence hereditary characteristics. So they're different versions of a gene. Each person inherits at least 2 alleles for a particular gene, 1 allele from each parent. If the 2 alleles are the same, then the individual is homozygous for that gene. If the alleles are different, then the individual is said to be heterozygous. Dominant alleles are expressed in phenotype even if they're paired with a different allele. And then recessive alleles are only expressed when they're paired with an identical allele. So someone may have one of their alleles that's dominant and the other one that's recessive. And the one that's going to show up, that's going to be physically expressed or behaviorally expressed, is going to be the dominant allele. So they're a carrier then for that recessive allele. And so when they have children, they're going to pass that on potentially to their child. So it may be that, for example, you have a parent who has the dominant allele of long eyelashes. So that's actually a dominant trait. And let's say that that's what they as the parent have. So because it's dominant, they have very long eyelashes, but they also have the recessive allele of short eyelashes. If they then mate with a partner who also has a dominant allele for long eyelashes and a recessive allele for short eyelashes, then there's the possibility that one of their children or more than one of their children is going to have recessive alleles, both recessive alleles, so that they're not going to have genotypically, they're not going to have any long eyelash dominant alleles. They're just going to have 2 recessive alleles so that when they grow and they're developing, they're going to have short eyelashes. We'll be looking at this in a little bit more depth here in just a moment, but I would encourage you to take a look at this in your textbook. Make sure that you understand dominant and recessive. I think many of you probably already do from your readings, but if you didn't read your book, make sure that you have. This is something that some of you may have learned about when you were in high school or in other classes, but it's maybe something that you haven't thought about in a while for some of you. So make sure that you go back and you review that so you understand the difference between phenotype and genotype and dominant and recessive. Another important term is incomplete dominance, and this is where the dominant gene does not completely suppress the recessive gene. I remember when I was in, I think, a biology class in college learning about incomplete dominance, and the first time I learned about it was in relation to plants. And I think this is probably the easiest way to think about it. Let's say that you have 2 plants. You have one that is red and you have one that is white. And the end result of the crossbreeding of these 2 is a pink flower. You certainly don't need to know the details of exactly how that happens, but let's take it a step further and let's think about people. Now, incomplete dominance is not that common of traits or in genetics. It is something that happens with some of our traits, but it's not with the majority of our traits. But it's very interesting for us to think about and then to see physically when it's manifested or behaviorally. So let's say, for example, we have a child whose parent has curly hair and then the other parent has straight hair. So the chance of the child having wavy hair, which would be that intermediate between curly and straight, that's the most likely. Now, there are some other traits that are associated with incomplete dominance, some diseases that maybe you've heard about before, like Sachs Disease or sickle cell anemia. And sickle cell anemia is described in your textbook a little bit. But so incomplete dominance, again, is where that dominant gene is not completely suppressing the recessive gene. So you're seeing a little bit of that recessive gene show up, and then you're also seeing that dominant gene. Another really interesting concept that's related to development is twinning. And I have twins, they're almost 8 years old, and my twins are dizygotic or fraternal twins. So they don't look identical. They look similar, just like siblings would, but they're also very different. And one of the ways that people know that they're twins, of course, is they know that they have the same age, but just by looking at them, they look developmentally about the same. They're almost the same height. They're also about the same physical size. But again, other than that, they just look like typical siblings. And this is because they are 2 unique individuals genetically. Now, The thing that's different, of course, about dizygotic twins is that they have a shared environment that siblings did not have in the womb. So they are more similar in their experiences than just typical siblings. And as you're going to be learning in this course, the environment prenatally is very important in affecting development. And so when dizygotic twins who've had that shared environment are growing and developing, that's going to create more similarities than you would see in, let's say, typical siblings. So dizygotic twins, what's happening here is you have two eggs or ova, and they're released and then fertilized by two separate sperm. And of course, fraternal twins, we're talking about two babies, but there are lots of instances where women will get pregnant with more than two babies. You may have We've heard of women who've, of course, you hear about triplets and quadruplets, and you've heard about women who've gotten pregnant with many more babies at the same time than that, and that's typically due to in vitro fertilization. But fraternal twins actually isn't all that uncommon. It tends to happen more frequently among people of certain ancestry. So it turns out that people who have Southern European descent, they're more likely to have dizygotic twins. And then, of course, like I mentioned, using some of the newer technologies. And when I say newer, I mean that have been developed in the past 20 to 30 years and are much more commonly used today in recent years than they were in the beginning when they were first developed. That's where we tend to see fraternal twins. And sometimes women, of course, are waiting to have children until they get older. We're seeing a lot of changes there in choices that women are making, where they may choose to go to college, then have a career, and then they may be waiting to have children, which then, as they get older, their fertility declines. And so they may seek out having some assistance with reproductive technologies. And I've also heard of some women who want to have twins, and so even though they're young or they've had no trouble getting pregnant, they will ask their doctors to give them medicine that will increase the number of eggs that are released. Now, that's an ethical issue that you have to think about, and not all doctors would, of course, be willing to do that because there are risks associated with having twins, with being pregnant with twins, that are unique risks that you don't have with a single pregnancy. And we'll be looking at that later here very soon in future course material. So dizygotic twins, again, they share the same amount of genetic material as their non-twin siblings. So my twin boys that are almost 8, they have a sister, and she is almost 4. She'll be 4 very soon. And her genetics is, she is just as unique as my twins are from each other. So they're all just like typical siblings. The only difference, though, is that my twins had that shared prenatal environment. Of course, too, as you learned in the last chapter, the environment that you grow up in at home very early on is unique as you're developing, based not only on factors that are related to things like, how you interact with your parents and parenting styles and things like that. But it's also unique based upon the time that you grow up. So my kids are both growing up at the same period of time historically. So when they were 1, the same things were going on for both of them that maybe aren't the same for my daughter, even though she's just 3 years younger than them, or 3.5 or so years younger than them. We could say that because of technologies changing so rapidly, that her experiences are probably going to be very different than theirs will be when she's their age. So we have to keep that in mind as well when we think about development. So monozygotic twins, those are twins that are identical. And as you can see in this image here, we have one ova that is fertilized by one sperm, and then that fertilized egg splits. And now we have 2 genetically identical people. Now, these individuals, when they're born, are going to look very similar, but I do want to point out that the environment, of course, starts to play a role the moment of conception. And while they do have that shared environment, just like the dizygotic twins did, something that's very unique about monozygotic twins is that they have to compete more so for resources and for space. So you sometimes will see that identical twins, although they may look the same, that one may be bigger than the other because one had greater access to nutrition than the other. And sometimes you might find that very sadly, if one twin is dominating tremendously, the other one may not be getting the resources that it needs and may not survive. But often what you might see, if we do see that disproportionate access to resources, you might see one twin that during pregnancy is developing a little bit faster, is a little bit bigger than the other one. And that's something that the OBGYN is going to want to keep an eye on to make sure that the other twin who may not be getting access to the same resources or the same amount, that they're developing and progressing as they need to. And then when those babies are born, sometimes you'll see that one is going to be bigger than the other. And eventually these two babies may catch up to each other, but sometimes they don't because access to nutrition in the womb affects physical development, including brain development. And interestingly, though, I will say that you can provide interventions for babies that didn't have the same access as their twin sibling in the womb that can help them to overcome that reduced access to resources. We'll be looking at that later on. It's very interesting. So these are the differences between monozygotic and dizygotic twins. And on the left-hand side, you see an image of two young girls. These are identical or monozygotic twins, mono meaning one and zygotic meaning zygote or fertilized egg. And then on the right-hand side, we see dizygotic twins. And so, of course, this is an example of a boy and a girl. You could have two girls, two boys, but they're genetically unique individuals. So let's take a look at some concepts that underlie genetic disorders. The first one is sex-linked, and this means that there's a defective gene on the X chromosome. And of course, this is more common in males if the gene is recessive. Some examples of this include hemophilia and color blindness. So here we have a male with his X and Y chromosomes, and here we have a female with two X chromosomes. So let's imagine that the male has an X chromosome for color blindness, and let's imagine that the female has one X chromosome for color blindness, and her other X chromosome does not contain color blindness. And so what we would see here, because there is no additional X chromosome, is we would see that there is nothing to dominate this recessive trait of color blindness. So we're going to see color blindness being expressed. And for this female, we're going to see that because this is a recessive trait and she has another X chromosome, we're going to see that she is just a carrier. So this is not going to be expressed. So she's not going to know that she's a carrier unless she has some genetic testing for some reason. Now, let's imagine that we have this same female here. And let's imagine that her mother is a carrier for color blindness. Her mother has no idea that she's a carrier for color blindness, but her dad is colorblind. And so when this girl is born, she will be born colorblind. She will have color blindness because she has both of these recessive characteristics or recessive X chromosome, or these recessive traits on her X chromosome. Let's take a look at another concept related to genetic disorders, and that's the concept of recessiveness. And so what we see is that this must be homozygous for this allele to be expressed. So some examples of this are PKU, cystic fibrosis, Tay-Sachs, and sickle cell disease. So we have what are called autosomes and sex chromosomes, and autosomes are the chromosomes 1 through 22, and then that last chromosome, the last pair of chromosomes, is going to be the 23rd one, and those are our sex chromosomes. So autosomal recessive inheritance, that's what we're talking about here, means that the gene in question is located on one of the autosomes. And so, like I mentioned, these are numbered in pairs, 1 through 22. So these autosomes don't affect an offspring's sex. Recessive means that two non-working copies of the gene are necessary to have the trait or the disorder. One is inherited from the mother and one from the father. If you only have one recessive gene, you're a carrier for the trait or disease. But if you don't have any health problems from carrying one, and you're not going to have any problems from carrying one copy of the gene. So most people, like I mentioned with sex-linked disorders, they don't know that they carry a recessive gene for a disease until they have a child with the disease or until they have a family member with the disease. And it's actually estimated that all people carry about 5 or more recessive genes that cause genetic diseases or conditions. So let's look at a few examples of these autosomal recessive disorders. One is cystic fibrosis, and this is a common inherited single gene disorder in Caucasians. People who have this disorder produce mucus that's very thick and sticky and can damage their organs. It can interrupt the function of vital organs, especially the lungs, and lead to chronic infections, and it leads to a shorter life expectancy. Sickle cell anemia. This is another common inherited single gene disorder, and it's found mostly in African Americans. About 1 in 500 African American babies are born with this, and about 1 in 12 African American people carries the gene for this disease. And so sickle cell disease involves the hemoglobin of red blood cells and their ability to carry oxygen. So as you may know, normal red blood cells are smooth, they're round, flexible, kind of like the letter O. They can easily move through your vessels in your bodies. But sickle cells are stiff and sticky, and when they lose their oxygen, they form into the shape of a sickle, or like the letter C. And they tend to cluster together and can't move easily through your blood vessels, and this can cause a blockage and stop the movement of healthy, normal, oxygen-carrying blood. And this is what causes sometimes the painful and damaging complications of sickle cell disease. Sickle cell disease can also affect the spleen, and when there is a problem with the spleen functioning, it's going to be very difficult for people to fight infections. People are more at risk for infections, and we know infants and young children are at risk in particular for life-threatening infections. So some of the treatments can involve emergency care for fevers and infections, being vaccinated to prevent to prevent yourself from getting sick to the point where you get these infections, penicillin, and then of course management of anemia. One more genetic autosomal dominant disorder that I'd like to mention, although there are several listed in your book, is Sachs disease. And this is normally a fatal disorder, it is a fatal disorder in children, usually by the age of 5. And this causes progressive degeneration of the central nervous system. And this is there's a particular enzyme that is absent in children who have this. And without this enzyme, a fatty substance builds up in the nerve cells, and in particular the brain. And then this, of course, does lead to death. So the process of this disease, it does begin early in pregnancy when the baby's developing, but it's not apparent until several months after birth. Right now, there's no cure for Sachs disease. And like the other ones I just mentioned, it does seem to be tied to ethnicity. About 1 in 27 people who are of European Ashkenazi Jewish ancestry carry the Sachs gene. Let's take a look at chromosomal abnormalities. This is when a child inherits too many or too few chromosomes. And as your book describes, maternal age is the most common cause. One example of a chromosomal abnormality is trisomy 21, also known as Down syndrome. And this is where there are 3 21st chromosomes. So some of the symptoms of trisomy 21 you can see at birth. So for example, this can include flat facial features, a smaller than normal head and ears, and a short neck. As the child grows, there will be some additional symptoms. Some of these could include a protruding tongue, a deep crease in the palm of the hand with relatively short fingers, white spots in the iris of the eye. Poor muscle tone is another one. Another symptom is small hands and feet. And there are some health conditions that are associated with trisomy 21. And these can include heart disease, congenital heart disease, some hearing problems, intestinal problems, celiac disease, eye problems like cataracts, thyroid dysfunction, skeletal problems, dementia, infectious diseases, and this is because of abnormalities in their immune system. Some mothers and fathers may know before birth that their child has trisomy 21 because they've already had genetic testing. Perhaps they had genetic testing because of some other risk factors. Maybe the mother is of advanced maternal age, which means that she may be 35 and older. Some women who are that age decide to do genetic testing so that if there are any genetic abnormalities, they're prepared for those in advance. And some mothers, like I said, may not know this. It's overall, regardless of whether they know this or not, it's very important for the mother, the father, the family to receive support. And this can be in the form of having healthcare providers, teachers, and therapists that the family works with, seeking out other families to help to provide them with support. Those who maybe have gone through similar experiences, this can be very beneficial. Some of these support groups might be in hospitals, physicians' offices, and schools. And I think it's very important that families don't believe the myths about Down syndrome. There have been many strides in recent years with people who have Down syndrome. Most live with their families and go to mainstream schools and have various jobs as adults. And people with Down syndrome can have very fulfilling lives. So I think that's a very important thing for families to remember. Sex-linked chromosomal abnormalities are abnormalities on the 23rd pair. A couple of examples of these are Turner syndrome and Kleinfelder syndrome. So with Turner syndrome, part or all of one of the X chromosomes is lost. It's a condition that affects only females, and it can cause a variety of medical and developmental problems. These can include things like short height, failure of the ovaries to develop, and heart defects. So Turner syndrome can be diagnosed before birth, prenatally. It may be diagnosed during infancy or in early childhood. So when we think about before birth, there are some symptoms that may be evident to the doctor. For example, when a mother goes in for her anatomy scan, which is normally around approximately 20 weeks or 19 weeks, where they look to see the development of the baby, they are going to look for things like, how is the heart functioning? How is the stomach functioning? Is there typical growth? And there'll be some signs that may be indicators of issues or problems. Those that indicate Turner syndrome can include having a large fluid collection on the back of the neck or other abnormal fluid collections, heart abnormalities and abnormal kidneys. Some of the features that a child who's born with this have, they may have swelling of the hands and the feet. They might be slightly smaller than average in their height at birth, have slow growth, cardiac defects. They might have a receding or small lower jaw, short fingers and toes. So you might also see a broad chest with widely spaced nipples and low set ears. And then in childhood and teens, some of the symptoms include slowed growth, not having growth spurts at expected times, adult height that's significantly less than might be expected for a female member of the family, failure to begin sexual changes expected during puberty, sexual development that seems to stall during the teen years, having an early end to menstrual cycles that are not due to pregnancy. And for most women who have Turner syndrome, they'll have an inability to conceive a child without fertility treatments. Let's take a look at Kleinfelder syndrome. With Kleinfelder syndrome, we have an extra X chromosome that's present in the cells of the male. So it is a genetic condition affecting males, and it's often not diagnosed until adulthood. Kleinfelder syndrome can adversely affect testicular growth, resulting in smaller than normal testicles, and it can lead to lower production of testosterone. It can also cause reduced muscle mass, reduced body and facial hair, and enlarged breast tissue. Some of the effects seem to vary, and not everyone has the same signs and symptoms. Most men who have Kleinfelder syndrome produce little or no sperm. But as with Turner syndrome, assistive reproductive technologies can be helpful so that the person with Kleinfelder syndrome can still have may be able to still father children. Let's take a look at genotype-environment interactions. These interactions involve genetic susceptibility to the environment. So each of you are going to be affected differently by the environment depending upon your genotype. So in this example, the effects of parenting style may depend on a child's genotype. So let's imagine that a child has inherited the genetic tendency to be very sensitive to the environment, to be very sensitive to environmental stimulation, to not deal very well with change. And let's imagine that their parents are not very thoughtful about this. Let's imagine that they're not very conscientious. Now, that's not an official type of parenting style. We'll be looking at different parenting styles, which include permissive, authoritarian, authoritative. But let's just think about this hypothetically. So the parent is not as thoughtful about how their behavior affects their child. And so the child who's very affected by environmental stimulation, change in their environment, loud sounds, different textures, tags on their clothes, that child is going to be much more negatively affected by, they're going to be much more negatively affected by that parent than, let's say, another child who isn't very affected by environmental stimuli, who doesn't seem to be very affected by things in their environment that involve change and stimulation. Another concept is epigenetics, and this is the study of DNA modifications that affect gene expression and are passed on when the cell divides. So here we see that the genes are switched on and off because of environmental factors. These environmental factors can be stress, nutrition, physical activity, working habits, smoking, and alcohol consumption. So there's increasing evidence that shows that these environmental and lifestyle factors influence these epigenetic mechanisms. In this video, you learned about genes. You also learned about mitosis and meiosis, genotype and phenotype, homozygous and heterozygous, and the traits of dominant and recessive. You also learned about some of the genetic disorders that may be due to a gene defect or some chromosomal disorders. You also learned about polygenic and incomplete dominance and the function of genetic counseling and why some people may seek genetic counseling. You learned about behavioral genetics and genotype environment correlations and genotype environmental interactions, and you also learned about epigenetics. In our next video, you're going to be learning about prenatal development. You're also going to be learning about methods that are used to assess prenatal development. I look forward to seeing you online.